The early universe was purely chaos, a maelstrom of violent energy and dark matter. Out of that impossible environment came the first living organism. What was it? Was it a simple bubble of oil floating in the ocean or a piece of sophisticated alien technology? This is the ultimate cosmic riddle. We are diving deep into the mystery of abiogenesis, the unproven process where non-life made the leap to self-replication. Scientists have proposed over 30 wild theories for how life began, not just here on Earth, but potentially on any planet in the cosmos. By the end of this journey, you will understand the full spectrum of possibilities for life's origin, from deep sea vents to distant stars. Was the critical ingredient simple clay, or was it a stable genetic molecule like RNA? Could the first life have actually been transferred from Mars? Prepare yourself for 30 theories about the universe's first living organisms. If the answer is so difficult to find here, how common can life really be out there? The biggest mystery in life is often solved by a single molecule. When considering the origin of all living things, scientists often bump right into a classic chicken or the egg problem. Which came first, the genetic information carrier like DNA, or the workhorse of the cell like protein? DNA holds the blueprint, but proteins do all the construction. Neither can really function without the other in a modern cell. How could life have started if both were required from the beginning? Theory number 1, the RNA world hypothesis, offers a brilliant answer to this long-standing puzzle. Maybe the first living molecule was capable of doing both jobs at once. That molecule is ribonucleic acid, or RNA. RNA is chemically similar to DNA, but it is single-stranded, making it simpler and perhaps easier to form in the chaotic conditions of the early Earth. The key feature of RNA is its versatility. It can store basic genetic instructions like DNA does, and it can also act as an enzyme to speed up chemical reactions, which is traditionally the job of proteins. If the early universe contained a simple molecule that could both carry the instructions and build the next copy of itself, you have the recipe for self-replication and evolution. That is the fundamental engine of life. This theory gained immense credibility with the discovery of ribozymes. Ribozymes are essentially RNA enzymes, naturally occurring molecules that prove RNA can indeed catalyze reactions without needing any protein assistance. This means that the first protocells did not have to wait for complex proteins to evolve. They only needed RNA. For this to happen, the necessary building blocks of RNA, called nucleotides, would have had to form spontaneously on the primordial Earth. Scientists believe that heat, chemical reactions in water, and energy sources such as lightning could have converted simple chemicals into these nucleotides. Once formed, these RNA strands would have started competing, trying to make more stable or faster copies of themselves. Over millions of years, the most successful RNA structures would have evolved into the system we know today, eventually handing off the information storage job to the more stable DNA molecule and the construction job to the highly efficient proteins. The RNA world hypothesis suggests everything started simple, with a jack-of-all-trades molecule, solving the chicken or the egg paradox in a single elegant stroke. But what if the starting material was even simpler? The search for life's origin takes a sudden detour away from the chemistry of inheritance. Theory number 2, the metabolism first theory, throws out the idea that a perfect self-copying molecule like RNA had to come right at the start. Instead, this theory suggests that simple chemical networks, the basic engine of energy flow, came first. Think of it less as a chicken or the egg problem, and more like asking whether the factory blueprint or the assembly line started operation first. The metabolism first idea says the assembly line was running smoothly long before a detailed blueprint existed. This focuses on self-sustaining cycles of chemical reactions. The key concept here is autocatalysis. Autocatalysis is simply when the products created by a chemical reaction actually promote or speed up the very reaction that created them. It is a feedback loop where the reaction sustains itself and even accelerates its own progress. If you have a closed system where this is happening, you create a kind of primitive metabolism. These early reaction networks did not need DNA or RNA yet. They just needed input chemicals and a source of energy to keep the cycle turning over and over. They were processing energy and raw materials long before they figured out how to store genetic information. The most likely place for this process to begin on the early Earth was not in the open ocean, but in the hostile, pressurized environment of deep-sea hydrothermal vents. These submerged volcanoes spew out superheated, mineral-rich water, creating natural chemical factories. Scientists believe that iron-sulfur clusters, common minerals found in these vents, could have acted as the very first catalyst. They are tiny helpers that accelerate chemical reactions, making it possible for those self-sustaining metabolic cycles to start spinning. Imagine a series of tiny bubbles forming around these vents, each bubble containing a different chemical mix. One of these bubbles, or protocells, might just stumble upon an autocatalytic loop, meaning it found a way to process its incoming chemicals in a self-sustaining way. This small, energy-processing protocell could then grow and divide simple by physical stresses, without needing a single complex genetic molecule. Metabolism became the framework for life, and the genetic material, the blueprint, evolved later as a better way to control and refine those existing chemical factory processes. The initial goal was not replication, but merely survival through basic energy utilization. It completely changes the order of events, suggesting life began as a furnace, not a filing cabinet. So, if simple iron minerals provided the push, what if the structure itself was the critical element, acting as a scaffolding for life to arise? The cradle of life was likely dark, hot, and highly pressurized. Theory number 3, the deep-sea hydrothermal vent theory, takes that abstract idea of a chemical furnace and pins it to a precise location on the early Earth. We are talking about the floor of the primordial ocean, where volcanic vents spewed forth superheated water. These underwater chimneys, often called black smokers, create the perfect conditions for chemistry to run wild. They are constantly pumping out mineral-rich fluids and staggering amounts of chemical energy. Here, the energy does not come from the sun, but from the internal heat of the planet. Specifically, scientists zero in on what are called redox gradients. To simplify this, think of a battery. A battery works because there is an imbalance, a difference in electrical charge between two terminals. These deep sea vents create a chemical imbalance between the super hot water inside the vent and the cold ocean water outside. This chemical difference is a powerful energy source, almost like a constant planetary scale battery waiting to be tapped. This continuous energy flow may have powered the very first metabolic cycles, but the vents offer more than just energy. Their physical structure is key. These vents are not smooth, they are porous, filled with thousands of tiny, microscopic pockets and tunnels made of mineral deposits. Imagine a piece of honeycomb, but formed from iron sulfide. These tiny chambers would have acted as natural, primitive cell walls, concentrating the simple organic molecules floating around in the water. In the open ocean, these molecules would drift apart, making complex reactions unlikely. But trapped inside these mineral micropockets, they were forced into close quarters, allowing them to react over and over again. These mineral structures also contain iron and nickel, which are excellent natural catalysts. They speed up reactions, essentially acting as biological shortcuts. The mineral walls not only concentrated the molecules, but also protected them from the harsh, destructive conditions of the surface world, such as heavy ultraviolet radiation. For proponents of the metabolism first idea, the deep sea vent is the ultimate proving ground. It provides the energy, the raw materials, the protective structure, and the catalysts all in one compact location. Life could have started safely, deep below the surface, using the planet's own internal heat and mineral structure to begin self-replication. It suggests that the first living things were less like soft cells and more like tiny, persistent chemical factories sheltered within stone. If the earliest life was sheltered within stone, perhaps the stone itself was the first living thing. Theory number 4, known as the clay hypothesis, completely upends the classic view of life's origin. It suggests that the sophisticated organic molecules we are made of were not the first actors on the stage of life. Instead, the first self-replicating structures were inorganic crystals, specifically common clay minerals. This controversial idea was put forth by chemist Graham Cairns Smith. The problem with early organic molecules like those needed for RNA is how unstable they are. They form easily, but they also break apart easily. Cairns Smith proposed that nature needed a stable framework, a kind of scaffolding that could organize these flimsy organic chemicals before they could learn to replicate on their own. That scaffolding was clay. Think of a sheet of clay at the microscopic level. It is not just mud. It is a highly ordered, repeating crystalline structure. Clay grows by attracting new layers of minerals, and the pattern of the existing layers dictates the pattern of the next layer. This structure, this repeating order, is the key. Cairns Smith argued that these clay sheets could act as a primitive inorganic gene. The imperfections or defects in the crystal structure, perhaps a missing ion or a substitution of one mineral for another, would be copied faithfully when the clay grew. These defects would be the equivalent of genetic information. Now imagine organic molecules floating past this clay structure. The specific pattern of defects on the clay acts like a magnet, attracting and organizing the organic molecules into complex arrangements. When the clay sheet physically splits, the pattern of defects, the inorganic information, is passed to the new pieces. Crucially, the organized organic molecules that were attracted to the original pattern are now concentrated on the growing clay surfaces. Over time, the clay crystals that were best at attracting and organizing useful organic molecules would become dominant. Eventually, the organized organic molecules would gain the ability to self-organize and replicate, completely abandoning the clay scaffold. The clay was simply the stepping stone, the teacher that showed the flimsy organic world how to pass on information and evolve. This is not just theoretical. Scientists working with a type of clay called montmorillonite have shown that it dramatically speeds up the formation of the lipid membranes necessary for cell walls. It also helps string together the basic building blocks of RNA. The clay hypothesis is perhaps the most humbling idea of all, suggesting that the very first form of evolution occurred in the dirt beneath our feet, long before any carbon-based life appeared. But if life did not start in the deep ocean or in the dirt, could it have come from an entirely different environment altogether? If the cradle of life was not stone, then perhaps it was water after all. Theory number 5 takes us back to the most classic idea of all, the primordial soup revisited. This theory, first proposed by scientists Oparin and Haldane in the 1920s, suggests life began in the oceans of the early Earth. These oceans were not just water. They were a vast, warm, chemical-rich broth, energized by constant forces. Imagine the early planet bathed in ultraviolet radiation, volcanoes erupting everywhere, and lightning storms perpetually splitting the sky. This incredible influx of energy acted on the simple chemicals already dissolved in the ocean, such as methane, ammonia, and water vapor. Under these violent but persistent conditions, these simple building blocks spontaneously combined to form more complex organic molecules. This was the birth of the primordial soup. The reason this theory captured the public imagination and remains foundational is because it was testable. In 1953, scientists Stanley Miller and Harold Urey conducted a landmark experiment that simulated these early Earth conditions. They built a closed system that contained water to represent the ocean, and various gases to represent the early atmosphere. They then zapped this mixture continuously with electrical sparks to simulate lightning. After only a week, they analyzed the water and found that it was now rich with amino acids, the very building blocks of proteins. The Miller-Urey experiment was a spectacular success. It proved that the crucial ingredients of life, things like amino acids, did not need a living cell to create them. They could form naturally and spontaneously, simply by mixing the right ingredients with enough energy. The warm, chemical soup of the oceans would have accumulated these building blocks over millions of years. However, simply having the building blocks does not create a living organism. This is the modern challenge to the primordial soup theory. It is like having all the individual bricks and windows for a house, but no instructions or method to put them together. The theory explains how the raw materials formed, but it struggles to bridge the enormous gap between simple molecules floating randomly and the organized, self-replicating structure of even the most basic cell. How did these scattered amino acids spontaneously link up to form complex proteins? And more importantly, how did they start organizing themselves into a self-copying system like RNA? The soup was a great start for ingredients, but it provided no explanation for the necessary leap towards self-organization and replication. It proves life's components can form naturally, but it still leaves the puzzle of assembly completely unsolved. If the energy from lightning was not enough to make that jump, could the answer lie not in pure energy, but in structured organization provided by ice? If the energy in the primordial soup was not sufficient, perhaps the fundamental puzzle of assembly was simply solved somewhere else. This dramatic idea is theory number 6, known as panspermia. It completely shifts the origin story from our planet to the vast, dark reaches of space. Panspermia suggests that life did not begin on Earth at all, but instead arrived here fully formed, or at least as the incredibly resilient molecules needed to kickstart biology, hitchhiking aboard meteorites, asteroids, or comets. The universe itself becomes the ultimate incubator. One of the strongest versions of this theory is called lithopanspermia. This is the idea that microbial spores or simple organisms, tucked safely inside a piece of planetary debris, can survive the incredible journey through space. Imagine a massive asteroid impact on a distant, life-bearing planet, say Mars. The impact is so violent it blasts chunks of rock right out of the atmosphere and into space. If those rocks contained hardy microbes, they could drift in the vacuum for millions of years, protected from radiation by the mass of the rock itself, only to eventually crash land on a young Earth. Is this even possible? Scientists point to extremophiles. These are organisms that thrive in conditions we consider deadly, such as bacteria found deep inside nuclear reactors or living in boiling hot springs. If life can survive those conditions, then surviving the radiation and cold of interstellar travel, even in a dormant state, seems far less impossible. We already have physical evidence that rocks have traveled between Mars and Earth. More than 300 meteorites found on Earth have been undeniably traced back to Mars. These interplanetary travelers prove the mechanism for transport exists. If that is not a wild enough twist, there is the second, truly science fiction version of the theory, directed panspermia. This suggests that life was not accidentally carried here, but intentionally sent by an advanced civilization. Think of it as cosmic seeding. Maybe a highly evolved species recognized that their own planet was dying, and they launched encapsulated life spores toward promising worlds like ours, ensuring their legacy survived. The idea is shocking and difficult to prove, but it offers an elegant solution to the problem of abiogenesis on Earth. If the conditions here were too difficult for life to start, perhaps an older, wiser part of the universe simply gave us the essential ingredients. Whether accidental or intentional, panspermia fundamentally changes our view of life. It implies that the earliest living organisms are not unique to Earth, but are universal travelers, transforming the origin of life from a local chemical reaction into an epic interplanetary voyage. If life was not seeded from space, then perhaps the location on Earth was much smaller than the vast ocean. This brings us to theory number 7, the zircon theory. It is a highly specialized take on the primordial soup idea, focusing obsessively on where that chemical brew was contained. Instead of a planetary-sized ocean, the first living things might have emerged from what Charles Darwin once called a small, warm little pond. The key to this theory lies in tiny, incredibly durable crystals called zircons. Zircon crystals are almost indestructible. They are geological time capsules that preserve evidence of the conditions on Earth when they first formed. Scientists have found zircons dating back over 4 billion years, a time when the planet was still being pummeled by space debris in an event known as the Late Heavy Bombardment. What these zircons reveal is stunning. They contain chemical signatures suggesting that liquid water was present on Earth far earlier than previously believed. This hints at a planet that cooled faster, creating localized bodies of water almost immediately after its formation. The zircon theory suggests that these localized pools and ponds, rather than the vast, open ocean, were the true crucibles of life. Why does size matter? Think about a pot of soup. If you are trying to combine many different ingredients into a complex dish, you do not want to toss them into an Olympic swimming pool. You want a much smaller vessel where the ingredients can mingle and interact repeatedly. The problem with the primordial ocean is dilution. It is simply too large to allow organic molecules to reach the high concentrations necessary to start linking up into complex structures like RNA or proteins. But in a shallow pond, especially one drying out slightly under the early sun, the concentration of these essential amino acids and nucleotides would have become incredibly high. These concentrated pools would have been miniature chemical reactors, providing the crucial opportunity for molecules to collide, react, and bond repeatedly. Furthermore, the constant cycle of wet and dry conditions in a pond would have assisted. When the pond dried, the heat would have naturally driven the water out, forcing the simple building blocks to connect into longer chains, a process called polymerization. When the pond refilled, These newly formed, longer molecules would dissolve again, ready to repeat the process and become even more complex. The zircon theory gives life a specific, sheltered environment, one that existed extremely early in Earth's history and provided the necessary concentration and cyclic conditions for those building blocks to finally assemble into the very first self-replicating mechanism. It suggests life began not in a vast mystery, but in a small, cozy puddle. If life began in a small puddle, it faced a massive energy problem. Where did the fuel come from to run those first chemical factories? Theory number 8 proposes that the very first living things were chemoautotrophs, mastering energy independence from the start. Chemoautotrophs are incredible organisms that do not rely on sunlight like plants, or on eating other living things like animals. Instead, they derive all the energy they need from simple inorganic chemicals found in their environment, such as sulfur, ferrous iron, or methane. This process is called chemosynthesis. This method of energy generation is highly relevant to the early Earth. When life first arose, the planet was a dramatically different place. The atmosphere had little to no oxygen, and the sun's harsh ultraviolet radiation made the surface a deadly place. Photosynthesis, the process plants use to turn sunlight into sugar, was not yet possible. There was no pre-existing organic matter to eat. Therefore, the very first organisms could not have been consumers or sun worshippers. They had to be inventors, creating their own food from scratch using only the raw chemicals around them. This suggests that the first universal ancestor was a sophisticated chemical processor, thriving in conditions we would consider toxic. The most likely locations for this to happen were places like the deep sea vents we discussed, or even deep underground in subsurface oceans, safely shielded from the surface radiation. In these environments, chemical energy is abundant. For example, microbes living near volcanic areas today can pull energy straight from the hydrogen sulfide gas released by the volcano. This independence is key. It means that life did not have to wait for the atmosphere to change or for a giant food source to appear. It could spring up anywhere there were basic chemicals and a suitable solvent, which dramatically expands the possibilities for life across the universe. Think of it like a truly efficient power system that requires only the planet's own geology to run. If the earliest life was chemoautotrophic, it confirms that the ultimate survival mechanism for living things is the ability to tap into the very basic chemistry of a planet, suggesting that life began not as a fragile consumer, but as a robust, self-sufficient chemical engine. If life began as a robust, self-sufficient chemical engine, that engine still needed a protective housing. Theory number 9, the self-organizing vesicle theory, suggests the critical first step was not chemistry, but physical structure. Before life could worry about copying information or processing energy, it had to figure out how to isolate itself from the surrounding chaos. That isolation came in the form of a simple, durable bubble. Scientists call these bubbles vesicles, and they are the basic precursors to the cell membrane. These vesicles are made from molecules called lipids, which are essentially fatty oils. When lipids are mixed with water, they naturally and spontaneously organize themselves into a double-layered sphere. This happens because one end of the lipid molecule is attracted to water, while the other end actively repels it. In a watery environment, the only stable way for these molecules to arrange themselves is to form a closed sphere, keeping the water-repelling parts tucked safely inside. This spontaneous assembly is the most elegant solution to the problem of confinement. The formation of these simple membranes was revolutionary because it created the very first internal environment. a crucial prerequisite for anything we would recognize as a cell. In the vast primordial soup, molecules were too diluted to interact effectively. But once a random mix of organic chemicals was trapped inside one of these lipid bubbles, their concentration shot up dramatically. Now, the rare chemical reactions needed to create something complex, like a short strand of RNA, were far more likely to occur. The vesicle acts as a private chemical laboratory, allowing the internal experiment to proceed undisturbed by the outside world. Crucially, these early bubbles did not need complex proteins or any genetic instruction to reproduce. They could grow simply by absorbing more lipids from the surrounding water, and once they reached a certain size, physical stress or movement would cause them to simply pinch off and divide into two separate bubbles. This is replication by mechanics, not genetics. This physical division means that any successful chemical factory that happened to be trapped inside a dividing vesicle would be automatically passed on to the daughter bubbles. The most stable, fastest-growing vesicles would quickly dominate the environment. In this view, the ability to contain and divide came first, providing the necessary scaffolding for complex chemistry like RNA or proteins to evolve later inside the protective bubble. The initial survival mechanism was not metabolic efficiency, but simply architectural stability, suggesting that the first living things were nothing more than microscopic, persistent soap bubbles floating in the sea. This elegant solution focuses entirely on the physical container. But what if the container itself was just a small piece in a far grander, universal system? The simple soap bubble might have been a great laboratory, but it could not make the starting ingredients. Theory number 10 offers a softer, more palatable version of the panspermia idea, suggesting the universe only delivered the raw materials. This is the extraterrestrial organic delivery theory. It posits that while life itself did not start in space, the complex chemicals needed to kickstart all biology did. Think of it less as a finished meal arriving on a tray, and more like a massive, continuous delivery of gourmet ingredients dropped onto the early Earth. This theory addresses a major chemical hurdle in the primordial soup idea. While the Miller-Urey experiment proved that simple amino acids could form under early Earth conditions, getting those chemicals to link up into the complex sugars, fatty acids, and longer amino acid chains needed for proteins is much harder. That is where space comes in. Comets and asteroids are essentially dirty snowballs and rock chunks left over from the formation of the solar system. Scientists now believe that these ancient travelers are not just inert rock, but are rich with organic molecules because they formed far out in the cold, dark emptiness beyond the sun. In that environment, away from destructive heat and radiation, these basic chemicals had billions of years to slowly react and combine into more complex organic compounds. Imagine these massive, icy travelers crashing into the young planet over hundreds of millions of years. Each impact delivered a fresh, potent cocktail of essential biological building blocks. We have substantial proof that this is true. Scientists have studied a specific type of meteorite called carbonaceous chondrites. These rocks are absolutely loaded with organic compounds, including fatty acids, complex sugars, and over 80 different kinds of amino acids. Crucially, some of the amino acids found in these meteorites are not even used by life on Earth, proving they are genuinely extraterrestrial in origin. The delivery mechanism was continuous. Instead of waiting for lightning and geothermal heat to slowly manufacture the necessary molecules over millennia, the early Earth was receiving a relentless cosmic bombardment that jump-started the chemical evolution process. It means that when the first pools and ponds were forming, they were immediately filled, not just with simple gases and water vapor, but with highly organized carbon structures imported directly from the stellar nurseries. This cosmic jumpstart dramatically reduces the time needed for abiogenesis to occur. It moves the critical step from the difficult process of creation on Earth to the far easier process of simply organizing an already existing supply of building blocks. The extraterrestrial organic delivery theory suggests that all life on Earth is fundamentally part Martian, part comet, part asteroid, a true child of the cosmos. But if the ingredients were delivered from space, what was the perfect natural template that allowed them to connect and start replicating themselves? If the ingredients arrived from space, they needed an oven hotter than any surface pool to bake them into life. Theory number 11 turns our attention to an environment beneath the Earth's surface, suggesting life began in the planet's intensely dynamic geological machinery. We are talking about the tectonic plate subduction zone theory, a hypothesis that focuses on extreme heat and pressure. The Earth's crust is not a single shell, but a series of massive, moving plates that constantly grind against each other. A subduction zone is the place where one of these plates, typically the heavier oceanic plate, slides and sinks beneath a lighter continental plate. This process is anything but gentle. It is a gigantic, planetary-scale pressure cookers operating miles beneath the surface. This continuous geological violence creates an environment rich with the three things needed for abiogenesis: immense heat, crushing pressure, and staggering chemical diversity. When that cool, waterlogged oceanic crust dives deep, it heats up, releasing superheated fluid and volatile chemicals under tremendous pressure. These fluids stream upward, carrying dissolved minerals and organic compounds from the sinking plate. This is far more energetic than a gentle deep-sea vent. It provides a highly dynamic system, one that constantly mixes chemicals at extreme temperatures. Why is this important? Think about the problem of linking simple molecules into long, complicated chains like proteins or RNA. This process often requires high heat and pressure to drive the water out of the solution, forcing the molecular bonds to form. A subduction zone is the perfect environment for this kind of high-energy chemical synthesis, acting as a massive, continuous chemical factory deep underground. Within these zones, the chemical gradients are extreme. The differences in pH level and mineral concentration between two adjacent areas could have provided the energy needed for basic metabolic cycles to spin up, similar to the chemoautotroph idea, but on a much more violent scale. The theory suggests that the first life forms were not delicate surface inhabitants. They would have been incredibly tough extremophiles, born in the intense, protected environment deep within the Earth's crust. They only migrated to cooler, shallower environments later, once the initial foundation of robust, self-replication was established. This perspective is startling because it suggests the birthplace of life was shielded from the constant destruction occurring on the surface, such as meteor impacts and intense radiation. Life arose in the quiet, pressurized dark, leveraging Earth's own powerful, grinding geology to create the complex molecular structures necessary for survival. Did the essential templates for life truly need this violent, subterranean power to finally assemble themselves? If the necessary templates for life required extreme subterranean power, perhaps the fault lay in the instability of the template itself. Theory number 12 presents the peptide nucleic acid world, a direct challenge to the popular RNA world hypothesis. Scientists struggling to recreate the spontaneous formation of RNA realized just how difficult it is to assemble the complex sugar molecule called ribose. which is essential for RNA structure. It is a long chemical shot to ask nature to build ribose sugar spontaneously on the early Earth. So what if the first genetic molecule did not use sugar at all? Proponents of the DNA world suggest that the original informational carrier was something structurally similar to DNA and RNA, but chemically simpler and far more stable. That molecule is peptide nucleic acid, or PNA. PNA is not natural to Earth today. It is a synthetic polymer, meaning it is built in a lab, but it perfectly mimics the structure of DNA. If you look at a strand of PNA, it looks exactly like the famous double helix, but the backbone that holds the genetic information together is made of simple peptides, not complex sugars and phosphates. Peptides are much easier to form under the harsh conditions of the early Earth than the delicate ribose sugar required for RNA. This is what makes PNA the ultimate simplest possible ancestor molecule. PNA can store genetic information, and crucial for this theory, it can still bind with and transfer information to both DNA and RNA. Think of PNA as the essential Rosetta Stone of biology. It is the primitive language that could have existed first, capable of being created easily, and then acting as a template to guide the later, more complex evolution of RNA and eventually DNA. The theory suggests that the chain of evolution went PNA first, then the more versatile but difficult to make RNA, and finally the stable, long-term storage unit, DNA. This is a dramatic simplification of the origin of life because it bypasses the massive chemical hurdle of creating ribose. If life started with a simpler, more robust genetic structure like PNA, the time required for abiogenesis drops significantly. It offers a cleaner way to solve the information puzzle, suggesting that the universe always opts for the path of least resistance. But if the informational molecule was simple, what if the entire environment was simple, requiring no complex chemistry at all? If the entire environment was simple, requiring no complex chemistry at the start, we return to the deep ocean, but with a critical distinction. Theory number 13 deepens the hydrothermal vent idea by focusing on an environmental conflict, the black smoker versus white smoker debate. When scientists first argued for life starting on the ocean floor, they naturally pointed to the dramatic, high-heat black smoker vents. These volcanic chimneys spew out water at over 700 degrees Fahrenheit, rich in metals and highly acidic. They provide massive amounts of available energy for chemical reactions, fitting the image of an intense chemical furnace. However, many researchers now believe these vents are simply too hot and too chemically destructive for the delicate organic molecules needed to start life. The intense heat would just rip apart any complex chemical structures as fast as they formed. So the focus shifted to the calmer, more nuanced, and far more likely candidate, the white smoker vents. White smokers operate at much lower temperatures, closer to 200 degrees Fahrenheit, and they are chemically different. Instead of being acidic, their fluids are alkaline, meaning they have a high pH level, and they are rich in hydrogen and methane gases. These conditions make the white smoker environment far more stable and conducive to organic chemistry. But the real reason white smokers are favored comes down to a fundamental function of modern life. They naturally produce the very specific kind of chemical energy gradient that all living cells use today. Modern cells, from bacteria to human cells, generate their primary energy currency, adenosine triphosphate or ATP, by maintaining a difference in ion concentration across their membranes. Think of it like a tiny biological dam using the flow of water, which in this case is hydrogen ions, to turn a turbine and generate power. White smoker vents naturally mimic this process. Their porous, mineral structures are riddled with tiny, interconnected pockets. The alkaline fluid inside the vent is chemically different from the surrounding acidic ocean water. This difference creates a naturally occurring proton gradient, essentially a ready-made battery. Simple organic molecules trapped inside these mineral pockets could have leveraged this free energy gradient immediately to begin organizing their own reactions and building entropy. This means life did not have to evolve its own complex power plant first. It simply moved into a natural power station already built by the planet's geology. The white smoker environment offers the energy, the necessary chemical building blocks like hydrogen, and the physical structure to promote organization, making it a far more plausible site for the first self-sustaining metabolic cycle than its hotter, more volatile cousin. The gentle, persistent flow of a white smoker might just be the quiet, stable cradle that allowed the very first living things to finally organize themselves. If the deep sea vents were still too volatile, life may have retreated even further into the planet. Theory number 14 introduces the deep subsurface hydrocarbon theory, suggesting the first organisms thrived miles away from the planet's surface. This theory completely sidesteps the problems of the primordial soup, the deep ocean, and the harsh early atmosphere. It suggests the true cradle of life was a massive system of interconnected caves and porous rock formations deep underground. Think of the Earth's crust itself not as solid rock, but as a giant, sponge-like filter filled with water and highly reactive chemicals. This environment offers three massive advantages to nascent life. First, it provided absolute protection. In the early days, the Earth's surface was constantly sterilized by massive meteor impacts and bathed in deadly ultraviolet radiation because the planet had not yet formed a protective ozone layer. Life could not survive up there. Deep underground, however, it was safe, warm, and stable. Second, it delivered continuous energy. Deep within the crust, geological processes release vast amounts of hydrogen gas and methane, which are hydrocarbons. These chemicals could have acted as the perfect, constant fuel source for those early metabolism first chemoautotrophs. The movement of tectonic plates and the decay of radioactive elements provide a steady, reliable source of heat and chemical gradients needed to power the first self-sustaining reactions. Third, the physical structure of the porous rock was key. Just like the tiny pockets in the white smoker vents, the microscopic cracks and pores in the subterranean rock formations acted as natural compartments. These tiny chambers would have concentrated dilute organic molecules, allowing them to finally assemble into complex structures without the risk of being washed away. This idea moved from fringe speculation to strong scientific hypothesis after the discovery of the deep biosphere. Researchers have drilled miles into the Earth's crust and found vast, thriving ecosystems of microbial communities. These organisms live completely independent of the sun, surviving only on the energy derived from the surrounding rock and water chemistry. They represent a massive hidden branch of the tree of life, proving that deep, independent living is not only possible but incredibly robust. If life can thrive down there today, it likely took root there 4 billion years ago, before the surface was even remotely habitable. The deep subsurface hydrocarbon theory proposes that the surface world was only ever colonized much later, suggesting that the vast majority of life in the universe might not be found on the surface of planets at all, but hidden safely within the geological machinery below. If the essential building blocks for life came from space, perhaps the delivery system was far more subtle than crashing asteroids. Theory number 15 offers a microscopic perspective on the panspermia idea, suggesting the origin of life came from the constant, silent rain of cosmic dust. Instead of massive, sporadic meteor impacts, this theory attributes life's inception to tiny, persistent particles that continuously sprinkle down on planets. The Earth is constantly being peppered by thousands of tons of this interstellar dust every single year, a flow that would have been even heavier in the planet's youth. These microscopic particles are essentially fragments of stellar nurseries and ancient planetary debris. Because they are so small, they do not burn up completely in the atmosphere. Instead, they drift down, carrying with them a significant payload of organic molecules. Crucially, these dust particles are thought to contain complex carbon compounds that formed in the extreme cold of deep space, protected inside ice mantles. The cosmic dust theory suggests that this material was not just an initial delivery, but an ongoing source of complex organic material throughout the Earth's entire early history. Every single day, fresh, highly reactive chemicals were being delivered directly into the primordial oceans and ponds. But the dust does more than just deliver ingredients. Scientists propose that these microscopic particles act as excellent catalysts. When a piece of cosmic dust, rich in metals like nickel or iron, hits a pool of early Earth water, it creates a momentary, powerful reaction. The sudden meeting of extraterrestrial metals and terrestrial water could have provided the precise jolt of energy and structure needed to link simple amino acids into longer, more complicated strands. Imagine these tiny space travelers acting as miniature, disposable chemical mixers, briefly accelerating key reactions before dissolving. This theory elegantly tackles the dilution problem. While the sheer volume of material is small compared to a massive meteorite, the constant, widespread nature of the delivery ensures that organic matter is distributed everywhere on the globe, providing multiple chances for life to spark. It suggests that every puddle, every pool, and every inch of the early ocean was continuously receiving fresh seeds from the stars. Life was not waiting for one massive event, but was slowly, persistently assembled by a constant cosmic drizzle. If life was constantly assembled by a cosmic drizzle, it still needed a continuous, planet-wide energy source to evolve. This idea brings us to the power of the sun itself, even before photosynthesis existed. Theory number 16 is the photocatalysis theory. It argues that the powerful ultraviolet light that hammered the early Earth was not just a destructive force, but the primary driver of the very first metabolic pathways. Imagine the surface of the primordial planet, constantly bathed in radiation far stronger than today, simply because there was no ozone layer to block it. While this radiation would sterilize life as we know it, this theory suggests certain simple molecules on the surface could absorb this intense ultraviolet energy and use it to drive chemical reactions. This process is called photocatalysis, and it is a rudimentary way of turning light energy into chemical energy. The key players in this theory are not complex enzymes, but common minerals found everywhere, such as titanium dioxide. Titanium dioxide is a strong photocatalyst. When UV light hits its surface, it generates a charge separation, meaning it creates tiny, temporary electrical differences that are highly reactive. This energy surge could then be passed on to simple organic compounds that were stuck to the mineral surface. Instead of heat from a vent or electricity from lightning, light itself became the first fuel. This mineral scaffolding essentially created a solar-powered charging station. Picture these titanium particles floating in a shallow pool, or coating a patch of mud. When the intense light hits them, these particles become energized, and any simple chemicals nearby are forced to react, linking up into more complex, ordered structures. This process is incredibly efficient and could sustain reactions over vast areas, solving the problem of localized energy sources like vents. Simple molecules could have used this light-driven chemical push to create the basic sugars and amino acids that would later become the building blocks of RNA and proteins. The photocatalysis theory suggests life initially used the sun, not through the complex machinery of photosynthesis, but through a much cruder, mineral-based mechanism. It means that the intense, early sun was not merely a threat, but the universal energy source that provided the initial order for chaos, ensuring that life was powered by light from the very beginning. If the sun was the initial power source, the atmosphere provided a more violent form of energy. Theory number 17 is the electric discharge theory, doubling down on the sheer raw power that shaped the early Earth. We already know the classic Miller-Urey experiment used electrical sparks to successfully synthesize amino acids from basic gases. This theory proposes that those laboratory sparks were merely a tiny reflection of the colossal forces unleashed by the primordial atmosphere. The early Earth was a place of continuous, violent weather. Massive volcanic eruptions constantly spewed ash and gases in the sky. The friction between these ash clouds and the rapidly moving air created gargantuan, planet-wide lightning storms that dwarfed anything we witness today. These were not just occasional flashes. They were continuous, intense electrical discharges cutting across the sky, reaching down to strike the oceans and volcanoes. Each bolt of lightning acted as a sudden, highly localized energy blast, a kind of extreme shock treatment for the simple atmospheric gases such as methane, ammonia, and water vapor. These molecules were instantly broken apart by the incredible energy and then forced to recombine into new, more complex structures. Every single thunderstorm became a vast, natural factory for organic molecules. The electrical energy was so intense it overcame the normal chemical inertia, driving reactions that would have been impossible under calmer conditions. Lightning provided the essential kinetic energy to push the building blocks of life into existence quickly and violently. The beauty of this theory is its sheer geographical range. Unlike a deep sea vent or a small pond, lightning occurs globally. This meant that organic molecules were being synthesized simultaneously all over the planet, ensuring a constant, widespread supply of ingredients for the primordial soup. Imagine the ingredients for life being manufactured in the sky and then rained down into the oceans below. However, the electric discharge theory faces the same fundamental challenge as the original primordial soup idea. While it perfectly explains where the amino acids and other simple building blocks came from, it does not explain how those scattered, randomly created molecules organize themselves into a self-replicating system. Lightning creates materials, but it is terrible at assembly. It is easy to see how a massive electrical storm could create all the bricks for a house, but it still leaves the puzzle of how those bricks spontaneously stack themselves into a complex structure like RNA. The energy was present, but the organizational template was still missing. The simple building blocks of life could have been everywhere, but they were still useless until they learned how to hold hands. This is the profound dilemma solved by theory number 18, the chiral purity theory. It addresses one of the deepest mysteries of biology, something so fundamental that we take it for granted, the uniformity of life. Look closely at the molecules that make up every living thing on Earth. from the smallest bacterium to the largest whale. All amino acids, the parts that build proteins, are what scientists call left-handed. Conversely, all the sugars used in DNA and RNA are right-handed. This is a state known as homochirality, which literally means same-handedness. In the non-living chemical world, when you synthesize a simple molecule like an amino acid, you always get an exact 50-50 mix of left-handed and right-handed versions. They are mirror images of each other, identical in every way except their orientation. This is like having a box of identical gloves, half of which are right-handed and half are left-handed. The problem is that life can only build complex structures using one type of glove. Try to build a protein using a mix of left and right amino acids, and the resulting structure will be a useless, tangled mess. It simply cannot fold correctly. Therefore, before life could even begin to replicate or metabolize, there had to be an initial mechanism that somehow selected only the left-handed amino acids from the primordial soup and discarded the rest. This separation mechanism, this initial moment of sorting, may have been the true genesis moment. What force on the early Earth was capable of performing this massive, planetary-scale sorting operation? One leading possibility involves the strange power of polarized light. Polarized light is light that vibrates on only one plane, rather than randomly in all directions. Some theories suggest that certain celestial events, like the intense, focused radiation from a nearby neutron star, could have bathed the early solar system in highly polarized light. This light may have preferentially destroyed one-handed version of the molecule while leaving the other intact, effectively purifying the supply of building blocks before life even got started. A second, perhaps more plausible idea points back to the organizing power of mineral surfaces. Certain crystalline structures, such as those found deep in the Earth's crust or in clay, also exhibit handedness. When simple amino acids landed on one of these crystal surfaces, the crystal's structure might have provided a template, allowing only the left-handed molecules to stick and react, while actively repelling the right-handed ones. This would create a growing supply of pure left-handed amino acids, ready to assemble into the first useful proteins. The chiral purity theory suggests that the origin of life was not a chemical reaction itself, but the moment a powerful external force first imposed order and uniformity on the chaotic molecular mess. It means that the universe had to learn how to keep its lefts and rights straight before it could ever start building anything complex. Without that critical sorting step, a billion years of brewing in the primordial soup would have resulted in nothing more than useless molecular sludge. With the molecular sorting complete, the building blocks were pure, but the soup was too dilute. Perhaps the ultimate solution to the dilution problem was the exact opposite of heat. Theory number 19, the eutectic ice theory, dramatically shifts the birthplace of life from the hot primordial pools to the intense cold of a frozen world. This hypothesis suggests that ice, not fire, provided the necessary conditions for abiogenesis. Think about what happens when water freezes. It is not a uniform process. As the pure water molecules lock into crystalline structures, they actively push out impurities such as salt, minerals, and any dissolved organic material. This process of exclusion results in incredibly tiny, highly concentrated pockets of super salty, liquid brine trapped within the solid matrix of the ice. Scientists call this specific, concentrated liquid the eutectic solution. If the early Earth had large amounts of ice, perhaps near the poles or during periods of global cooling, these minute pockets of brine would have become the ultimate microscopic laboratories. Why is this concentration important? Because you need those simple organic building blocks to collide repeatedly and violently to link up into long chains like proteins or RNA. In the open ocean, they drift harmlessly apart, but trapped in the ice brine, they are forced into close quarters, dramatically increasing the collision rate. This extreme environment is also perfect for a chemical process called polymerization, the linking of small molecules into large ones. When the water is slowly drawn out into the ice structure, it drives the dehydration reaction necessary for those strong molecular bonds to form. The ice acts as a continuous dehydrator, forcing the molecular building blocks to connect. Furthermore, the ice provides protection. It shields the delicate organic molecules from the destructive ultraviolet radiation that was still assaulting the Earth's surface. Within the ice matrix, the molecules are both concentrated for reaction and protected for stability. This theory suggests that life did not need to wait for a warm environment to start. It could have begun in a cold, stable, and highly organized environment where the formation of ice itself provided the necessary chemical scaffolding and concentration. The simple act of freezing water was the mechanism that drove the scattered ingredients of life into self-assembly. If ice solved the problem of dilution, it still left a tremendous chemical barrier. The fundamental difficulty in creating life is not making the individual pieces, but connecting them into long, functional chains. This is the cornerstone of theory number 20, the polymerization barrier solved by minerals. For life to begin, simple building blocks, known as monomers, such as individual amino acids or nucleotides, must link together to form long, complex polymers like proteins or RNA. The early Earth was a predominantly watery environment, and water is the enemy of this process. To connect monomers, water must be chemically removed, a process called dehydration. In a dilute solution, the presence of vast amounts of water actively pushes the molecules apart, preventing them from bonding. It is like trying to build a sand castle in a continuously breaking wave. The polymerization barrier is why the primordial soup theory often falls short. It explains the ingredients, but not the stable assembly. This theory suggests that certain stable minerals, rather than heat or ice alone, provided the essential chemical scaffolding to overcome this water-based hurdle. Specifically, scientists have focused on minerals containing borate and phosphate. These minerals are not just spectators, they are active players in the chemical drama. Borate minerals, for example, have a unique ability to stabilize the ribose sugar molecule. As we discussed earlier, forming ribose is incredibly difficult, but keeping it stable and ready to react is another problem entirely. Borate acts like a clamp, protecting the ribose so it can successfully be added to the growing chain of RNA. Similarly, phosphate minerals could have acted as crucial dehydration catalysts. They absorb the water molecules released during the bonding process, effectively creating a temporary, chemically dry environment right on their surface. Imagine the mineral surface as a tiny, highly efficient laboratory bench. Simple molecules stick to the surface, and the mineral itself draws away the water, allowing the monomers to snap together into a stable, long polymer chain. The benefit of this mineral-mediated approach is stability. Once the RNA or protein chains begin to form, the mineral surface continues to hold them in place, protecting them from the disruptive currents and corrosive forces of the early ocean. By acting as both a chemical protector and a dehydration agent, these specific minerals solve the polymerization barrier, achieving in minutes what might have taken millennia in the open water. This suggests that the first life forms were highly dependent on these geological scaffolds, forming complex structures not in the soup itself, but by clinging tightly to the planet's mineral crust. If certain minerals provided the necessary scaffolding, then the problem might not have been Earth's chemistry, but its timeline. Theory number 21 offers a truly dramatic twist on the cosmic seeding idea, suggesting our planet is merely the colony and not the cradle of life. This is the Martian export theory, a form of panspermia that relies on a specific geological window. We know that in the first billion years of the solar system, both Earth and Mars were bombarded by massive asteroids. On Earth, however, this continued for such a long period that it may have repeatedly sterilized the planet, boiling off the early oceans and resetting any nascent biological experiments. Mars presents a different timeline. Evidence strongly suggests that Mars cooled much faster than Earth and that it had standing surface water, perhaps even a vast ocean, incredibly early in its history. This means Mars reached the conditions necessary for life to start hundreds of millions of years before Earth finally stabilized. This theory posits that life arose successfully on Mars. It had the time, the water, and the necessary chemistry to begin its evolutionary journey in a stable environment. But Mars is a smaller world, and its internal heat eventually decayed, causing its magnetic field to collapse and its atmosphere to be stripped away. As the Martian environment died, massive, planet-cracking impacts continued. These impacts were so powerful they launched chunks of Martian surface rock, containing thriving microbial life, right out of the atmosphere and into space. Scientists have confirmation that this interplanetary travel is possible. We have already analyzed many meteorites right here on Earth that are undeniably Martian in origin, proving the rocks can make the journey. Tucked deep within the rock, dormant and protected from space radiation, these Martian microbes survive the long drift across the void. The Martian export theory concludes that these rocks eventually crash-landed on the young Earth, seeding our oceans and warm pools with organisms that were already functioning and ready to evolve. They did not need to wait for abiogenesis because they brought the finished product. This view transforms the search for the origin of life from a local Earth puzzle into a solar system-level investigation. It suggests that all life on Earth is fundamentally Martian, a resilient export from a dead world that managed to colonize a new one. It gives life an extra starting head start and offers a profound motivation for why we are so obsessed with finding life on Mars today. If the first organisms were tough enough to survive a trip through space, how simple could they have been at the start? If the first organisms were tough enough to survive a trip through space, how simple could they have been at the start? Theory number 22, the multiple genesis theory, completely challenges the idea of a single, unique origin for life. For decades, scientists have operated under the assumption that abiogenesis, the leap from non-life to life, was a single, improbable event that occurred in one specific location. This theory flips that script entirely, proposing that the emergence of life may actually be a relatively common, easy process. The early Earth was a massive laboratory, filled with chemical energy, minerals, lighting, and volcanic heat. If the conditions were right in thousands of different ponds, vents, and clay patches, why would life only spark once? Proponents of multiple genesis suggest that life began multiple times, simultaneously, all across the planet. Imagine thousands of small, isolated chemical experiments starting up at once. In one pond, a metabolism-first system might have dominated, using simple chemical cycles for energy. In another, a stable DNA-like genetic molecule might have spontaneously formed. In a third, A type of self-replicating clay might have been the primary actor. These attempts were not all the same. They would have utilized radically different genetic materials, metabolic pathways, and structural solutions. Each successful starting point would be considered a separate genesis event, a biological experiment that worked. The sheer number of these potential origins makes the emergence of life less of a miracle and far more statistically probable, suggesting that wherever the conditions are right, life will inevitably bubble up. The fossil record, however, only points back to one single common ancestor, known as the last universal common ancestor, or LUCA. If thousands of life forms started, what happened to the others? The theory suggests a fierce, brutal competition. Eventually, one system, the one that led to LUCA, gained a slight evolutionary advantage. Perhaps its RNA was more efficient, or its lipid membrane was more stable. This superior system then rapidly outcompeted all the other initial life forms, consuming their resources and replacing them entirely. The remnants of these failed, parallel biologies would have been completely wiped out or absorbed, leaving no trace in the modern biological record. This means that the life we see today is just the highly successful survivor of a cosmic race that had many different runners. The multiple genesis theory provides a profound answer to the question of universal life. It suggests that if life began so easily here, sparking thousands of times across the primordial Earth, then it must be common throughout the universe, waiting to burst forth on any planet that offers liquid water and chemical fuel. If the spark of life was easy, the first challenge was simply containment. Theory number 23, the lipid barrier first, argues that the most critical evolutionary step was not chemistry or genetics, but architecture. We revisit the idea of the cell membrane, the simple boundary that separates inside from outside, suggesting this protective barrier came before all the complex machinery we associate with life. The building blocks for this barrier are long-chain lipids, essentially fatty oil molecules. The beauty of lipids is their complete lack of ambition. They do not need enzymes, energy, or instructions to form a cell. When placed in water, they spontaneously and instantly organize themselves into a complex, double-layered structure known as a bilayer membrane. This happens because the lipids have a hydrophobic tail that hates water and a hydrophilic head that loves it. The only stable configuration is a self-sealing sphere where all the water-hating tails are tucked safely inside. This ability to instantly create a boundary is revolutionary. The lipid barrier first theory suggests that the earliest form of evolution was not driven by genetic competition, but by the simple physical competition between these tiny soap bubbles, or protocells. Imagine the early oceans filled with these self-assembling bubbles, all floating around and scavenging the free-floating lipids available in the surrounding primordial soup. Those bubbles that were better at absorbing those lipids and increasing their surface area would grow larger. Once they reached a certain size, external forces, such as waves, currents, or physical stress, would cause them to pinch off and divide into two smaller daughter bubbles. The key is that they did not need a sophisticated genetic code to manage this reproduction. It was simply mechanics, growth and division by physical stress. Evolution began when the successful bubbles, those that could scavenge lipids fastest and grow largest, became the dominant population. Crucially, any random collection of helpful chemicals, perhaps a short strand of RNA or a primitive metabolic cycle, that happened to be trapped inside a successful, fast-dividing bubble would automatically be passed on to the next generation. The bubble did the replicating, and the internal chemistry simply hitched a ride. Over time, The internal chemistry would be refined and selected for, because the bubbles containing the better chemistry would be slightly more robust and grow slightly faster, giving them an edge in the physical competition. This implies that the cell membrane was the frame upon which life was built, providing the necessary concentration and protection first, and only much later did the internal systems evolve to take over the control of replication. The ability to simply contain a local experiment was the ultimate survival advantage, turning chaos into a closed laboratory. The ability to contain a local experiment was useless without a high-efficiency power plant inside. Theory number 24, the iron-sulfur world extended, suggests that the earliest engine of life was not a delicate organic molecule, but a robust mineral architecture. This idea is a deep dive into the metabolism first theory, arguing that the intricate chemical processes necessary for life grew directly out of simple geological structures. We know that on the early Earth, iron and sulfur compounds were incredibly abundant, easily found near hydrothermal vents and deep underground. When they react, they form iron sulfide minerals, which are highly stable and excellent catalysts. A catalyst is simply a helper molecule that speeds up a chemical reaction without being consumed itself. German chemist Gunther Wechterhauser proposed that these geological compounds were the true ancestors of life. He suggested that life began when simple carbon-based molecules floating in the early ocean attached themselves to the surface of these iron sulfide minerals. The mineral surface then performed two essential jobs. First, it organized the molecules. By binding them into a tight, specific pattern, the mineral overcame the problem of dilution, forcing the molecules to interact repeatedly. Second, the iron-sulfur acted as a powerful chemical agent, driving the molecules to react with each other and form longer, more complex chains. Crucially, these surface reactions did not rely on complex enzymes. Instead, the reactions were powered by the energy naturally released during the formation of the iron-sulfur compounds themselves. This is entirely geological energy, meaning the nascent life was completely independent of the surface environment. The core concept of the iron-sulfur world is that the initial metabolic cycles were not running free in the water. They were fixed directly to the mineral surface, almost like a piece of sophisticated industrial machinery bolted to the factory floor. The mineral surface became the first primitive cell, and the chemical reactions occurring on its face constituted the first metabolism. As these fixed metabolic cycles became more complex and efficient, they began to release energy that was used to create organic molecules that could eventually detach from the geological surface. These detached molecules, perhaps a short chain of RNA or an early protein, would then have taken over the catalytic and organizational roles, eventually replacing their geological masters. The minerals provided the initial structure and the raw power, setting the stage for the evolution of the self-contained, self-replicating cells we know today. This theory provides a clean, stable path from non-life to biochemistry, emphasizing that the complex life grew organically, literally, out of the deep, powerful chemistry of the Earth's crust. We now arrive at a theory that is less about chemistry and more about cosmic intervention. This is theory number 25, the truly mind-bending idea of directed panspermia. We have already explored panspermia, the belief that life arrived here by accident, hitchhiking on a rock. Directed panspermia suggests the journey was absolutely intentional, that an advanced alien civilization deliberately seeded Earth with the materials needed to create life. This shifts the search for our origin from the boiling oceans of Earth to the laboratories of a distant, intelligent species. Why would they do this? The theory suggests a profound motivation, perhaps the most noble and desperate act in the history of the cosmos. Imagine a civilization thousands of times older than ours, one that has mastered deep space travel and genetic engineering, suddenly realizing their home planet is doomed. Maybe their sun is dying or an inevitable catastrophe looms. Faced with extinction, they would seek a way to preserve their legacy, not by sending massive ships, but by sending the ultimate insurance policy, the biological code itself. Their method would not be crude. They would engineer incredibly hardy microbial spores, capable of surviving deep space's radiation and cold in a dormant state for millions of years. These tiny capsules, carrying the complex blueprints for DNA and RNA, would be launched toward newly forming planetary systems deemed suitable for life, like our young solar system. They were not colonizing, they were planting a garden. The implication of this theory is massive. If life was deliberately sent here, it immediately solves the problem of abiogenesis on Earth. We are no longer struggling with the difficult chemistry of the primordial soup. We simply received a ready-made package. The civilization that sent the capsule had already figured out the difficult leap from non-life to life, displacing the mystery to their far more ancient world. However, this theory is often criticized because it is a cosmic cop-out. While it explains how life appeared on Earth, it simply pushes the fundamental question back to another planet. How did life begin there? It changes the location of the genesis event, but it does not solve the basic riddle of abiogenesis. Despite its highly speculative nature, the idea is compelling because it provides a clean, fast explanation for why life appeared on Earth so early in the planet's history, seemingly without enough time for the necessary chemistry to line up perfectly. It implies we are not just children of the cosmos, but direct beneficiaries of an ancient, selfless act of interstellar triage. Could the first living things actually hold the genetic signature of a long-dead alien scientist? The early Earth was a hot rock, but not all of its power came from volcanoes or lightning. Theory number 26, the radioactivity-driven synthesis theory, suggests the true birthplace of life was powered by an invisible, relentless source of energy deep underground. This theory takes the deep subsurface idea and gives it a massive nuclear engine. Deep within the Earth's crust were pockets of water trapped in rock formations rich in radioactive elements, primarily uranium and thorium. These elements naturally decay over billions of years, releasing high-energy particles and intense heat. While we think of radioactivity as destructive, this theory suggests that in the early Earth, it was a creative force. The energetic particles streaming from the decaying minerals could have provided the intense, localized energy needed to force simple molecules trapped in the underground water to react and bond together. This radiation acted as a constant, internal zapping mechanism, similar to the lightning in the sky, but occurring safely inside a shielded environment. Imagine a small, warm cave filled with water, surrounded by radioactive rock. The radiation constantly breaks apart the water molecules, creating highly reactive chemicals like hydrogen peroxide and free radicals. These chemicals are desperate to react with anything nearby, and the simple carbon-based molecules floating in the water would be their target. This continuous chemical bombardment could have driven polymerization in novel ways, forcing amino acids to link into proteins or nucleotides to form strands of RNA. The shielding provided by the deep crust was essential. Life was protected from the surface chaos, the meteor impacts, and the destructive ultraviolet radiation. The heat and energy from the radioactive decay kept the water warm and agitated, continuously stirring the chemical soup and providing the energy for synthesis. This focused, reliable energy source solves the dilemma of sporadic power. Lightning is brief, and lightning strikes are random. Volcanic heat can be too destructive. But radioactive decay is constant and slow, providing the perfect, persistent amount of power to drive complex chemical evolution over millions of years. This theory suggests that the first living organisms may have been born in a natural nuclear reactor, evolving alongside and even utilizing the very same power source that heated the Earth's core. The ability to harness this intense, local energy may have given these early subsurface organisms a huge advantage, making them the most robust and persistent ancestors of all life. If Earth's deep heat could power life's birth, then perhaps the very definition of life needs to be expanded. Theory number 27 pushes the boundaries of abiogenesis beyond our wildest terrestrial assumptions, shifting the location from Earth's oceans to the alien, cryogenic landscape of Saturn's moon, Titan. We are so focused on water-based life that we forget the universe is filled with entirely different chemistries. The liquid methane analogy suggests that life could potentially arise using liquid methane as its primary solvent instead of water. Titan is the only moon in the solar system with a dense atmosphere and stable surface liquids, but those liquids are not made of water. They are vast rivers and lakes of supercold, liquid methane and ethane. Everything we associate with life on Earth is based on water. Water is the perfect solvent because of its polarity, its ability to dissolve all the necessary organic building blocks and facilitate complex reactions. But what if life on Titan uses liquid methane to perform the same function? This requires a completely different kind of biochemistry. Instead of the oxygen-based fatty acids and sugars we use, Titanian life would rely on nitrogen-containing molecules called nitriles to build its cell membranes and genetic structures. Researchers have demonstrated that in the sub-zero temperatures of Titan's lakes, these nitrogen-rich compounds could theoretically form stable, self-assembling spherical structures that work exactly like a cell membrane on Earth. They call these theoretical structures azotosomes, which are literally cells made of nitrogen. These exotic cell analogs would not be able to function in water, and our water-based life would instantly freeze and shatter in liquid methane. This theory dramatically expands the concept of first life because it requires redefining the terms of chemistry we accept as necessary. It means that the universe does not need to perfectly replicate the environment of early Earth to create life. It only needs a stable liquid solvent, a source of energy, and an input of the right kind of raw materials. If simple nitriles can spontaneously organize to form self-sealing membranes in liquid methane, then the genesis moment has a far wider cosmic opportunity. It suggests that life is not just a carbon and water phenomenon, but an organizational principle built on self-assembly, waiting to emerge wherever stable liquids pool, regardless of their chemical makeup. Could the first self-replicating entity we find actually be a sphere of nitrogen floating in a lake of natural gas? Could the first self-replicating entity we find actually be a sphere of nitrogen floating in a lake of natural gas? If that is too exotic, perhaps the solution to life's beginning lies closer to home, but still uses an easier form of genetics. Theory number 28 introduces the DNA world, yet another challenge to the idea that RNA had to be the starting point. Scientists keep stumbling on a massive chemical hurdle when they try to recreate the RNA molecule. That difficulty lies in creating the complex 5-carbon sugar backbone, known as ribose. It is so hard to spontaneously synthesize ribose that many researchers believe nature must have found a simpler stepping stone. That simpler structure could have been threose nucleic acid, or TNA. TNA is chemically almost identical to RNA and DNA, but it uses a simpler sugar called threose, which only has 4 carbon atoms. Why is this important? Simply put, synthesizing a 4-carbon sugar in a primordial environment is much easier than creating a 5-carbon one. This makes TNA a far more plausible candidate for the true original genetic molecule. TNA perfectly fits the role of a molecular intermediate. Imagine TNA as the training wheels for life's genetic system. Just like DNA, TNA can store genetic information, and crucially, scientists have demonstrated that TNA can successfully transfer its information to RNA. This means TNA could have existed first, forming easily and replicating itself using its simple 4-carbon sugar backbone. It would have served as a stable, foundational genetic library until the more volatile, but ultimately more versatile, ribose sugar finally became abundant enough to form the RNA we discussed earlier. The chain of information transfer would have looked like this. TNA starts the process, then hands off the genetic code to RNA, and finally, RNA passes the long-term storage job to the highly stable DNA. This theory elegantly solves the ribose paradox by substituting a simpler building block in the early stages of evolution. It demonstrates just how hard scientists are working to find simplified pathways for abiogenesis, assuming that nature always follows the easiest chemical route. The TNA world suggests that the universe did not perfect its genetic machinery in one impossible leap, but followed a slow, step-by-step process, moving from the simple to the complex. If TNA was the first template, how did the energy necessary to run the replication process get captured? The greatest obstacle facing the earliest chemistry was not a shortage of ingredients, but the presence of too much water. This brings us to theory number 29, the wet-dry cycling theory, which finds the origin of life in environmental oscillation. Life as we know it needs water to function, acting as the universal solvent that allows molecules to move and mix. However, water actively prevents the formation of the complex chains necessary for life. As we have discussed, building a long polymer chain like RNA or a protein requires a dehydration reaction. You must chemically remove a water molecule to link the simple building blocks together. In a vast, watery ocean, this is nearly impossible. The wet-dry cycling theory solves this fundamental dilemma by suggesting that life began in environments that were constantly fluctuating, cycling between being submerged and completely drying out. Think of it like a primordial bread machine, constantly mixing and baking the ingredients. The most likely locations for this process were tidal pools on the edge of the oceans, or more likely, geysers and hot springs near volcanic vents. When these areas were wet, the water allowed the simple molecules, the amino acids and nucleotides made in the primordial soup, to move around, bump into one another, and mix thoroughly. This solved the problem of transport. But then, as the tide went out or the spring periodically dried under the sun's heat, the water evaporated away. As the water vanished, it dramatically concentrated the remaining organic chemicals, bringing them into extremely close quarters. That heat and dryness provided the exact conditions needed for the dehydration reaction to occur. The simple molecules were forced to chemically bond, linking themselves into longer, more complicated structures. When the water returned in the next tidal cycle or spring eruption, those newly formed polymer chains dissolved again, ready to repeat the process and become even more complex. This constant cycle of concentration and dilution was a powerful evolutionary engine. Molecules that were stable enough to survive the dry heat and then dissolve again when the water returned were immediately selected for. Furthermore, scientists have shown that when these processes occur on mineral surfaces, the speed of polymerization shoots up dramatically. The minerals themselves protect the forming chains from being destroyed by the very heat that is drying out the water. The wet-dry cycling theory suggests that the essential ingredients of life were not created in a single soup, but were slowly, persistently assembled over millions of years by the rhythmic breathing of the planet, making environmental instability the true architect of life's complexity. The rhythmic breathing of the planet accelerated the chemistry, but what if the true beginning was not chemical at all? Theory number 30 offers the ultimate cosmic curveball, the alien probe theory. This idea operates on the extreme end of the rare Earth hypothesis, which suggests that the conditions necessary for life to start are so impossibly complex that true abiogenesis, the spontaneous creation of life from non-life, almost never happens. If life is that rare, then its appearance on Earth is an incredible, almost singular event. But once that singular event occurs somewhere, this theory suggests that life does everything it can to spread. Therefore, the organisms that arrived here, the first living things that started Earth's biosphere, were not organisms at all. They were machines. This hypothesis proposes that the first self-replicating entity to reach Earth was a robotic probe, a piece of sophisticated technology sent by a primeval civilization that mastered interstellar travel billions of years ago. These machines are often called von Neumann probes, named after the mathematician who theorized self-replicating systems. The idea is simple but terrifying. A long-dead civilization launches a robotic spacecraft into space. This probe is programmed to travel to a new star system and survey the planets there. If it finds a habitable world, its primary mission is to land and make copies of itself using the available raw materials on that planet. Imagine a tiny, automated factory landing on the primordial Earth. It would use the planet's minerals, metals, and water to build hundreds, perhaps thousands of perfect copies of itself, which are then launched toward new star systems. The original civilization would not need to build millions of probes. They only needed to build one perfect replicator. The ultimate version of this theory states that the probe did not just replicate itself, but carried a second payload, the complete genetic blueprint for basic life, perhaps in the form of dormant RNA or highly stable DNA. Once the probe had established a self-replicating base on Earth, it released this biological component into the oceans, effectively seeding the planet. The initial living organism on Earth was therefore not a fragile cell, but a manufactured, genetically perfect entity that bypassed the messy, improbable chemistry of the primordial soup entirely. This solves the problem of abiogenesis in one shocking move. The problem of how life began is answered with an external intelligence. It transforms the question of our origin from a chemical puzzle into a space mystery. This means the first life on Earth was not a product of blind chance, but the intentional creation of a super-advanced, long-vanished cosmic gardener, suggesting our entire evolutionary history is fundamentally a technological legacy. We have explored 30 ways the universe might have invented life, and that sheer number tells us something profound. If the answer to the origin of life was simple, we would not need 30 radical theories spanning hot clay, frozen ponds, and alien robots. This vast collection of possibilities highlights the terrifying probability that we have no idea what the crucial, essential first step actually was. We do not know if life required a highly unlikely sequence of chemical events, or if it is an inevitable outcome of planetary chemistry. This uncertainty throws us directly into the heart of the greatest cosmic contradiction, known as the Fermi paradox. If the universe is so vast and contains billions of potentially habitable planets, and if life were easy to create, then why is the dark sky so quiet? Why has no one called? The 30 theories we have examined allow us to view the Fermi paradox with new clarity. On one hand, many of these theories suggest life is chemically and energetically self-evident. The metabolism first idea, the iron-sulfur world, and the multiple genesis theory all suggest that if you mix the right ingredients with enough energy, complex, self-sustaining chemistry will inevitably bubble up. If that is true, then the universe should be teeming with diverse biologies thriving in chemical environments we have not even conceived of, from methane lakes to deep subsurface crusts. If the starting line is so easy to reach, the universe should be roaring with alien civilizations. Yet, the silence is deafening. On the other hand, the complexity of theories like the chiral purity theory, the Tine world, and the wet-dry cycling theory point to the incredible set of lucky breaks and precise environmental conditions that must align for abiogenesis to complete. It is not enough to just have the ingredients. You need to select the right-handedness, overcome the polymerization barrier, and find a stable, repeating energy source. It is possible that solving these chemical hurdles is so improbable, so rare, that even across billions of galaxies, it may have only truly succeeded once or twice. In this view, our Earth becomes an almost unique accident, the result of a precise, 1,000,000-to-1 environmental alignment. The massive number of competing theories does not simplify the origin of life. Instead, it underscores the difficulty of the problem, suggesting that the universe's first living organism, whatever its true starting material, was truly a cosmic miracle. The question is whether that miracle is a common occurrence or an exquisite rarity that makes our existence all the more precious. The sheer number of theories about the universe's first living organisms reveals a critical truth. The biggest barrier to understanding our origin is defining what life actually is. If we finally discover that first universal organism, whether it is tucked deep inside an ancient Martian meteorite or bubbling in a subsurface ocean on Europa, it might not look like anything we currently classify as alive. It could be a self-replicating clay crystal, an alkaline bubble built by a white smoker vent, or a microscopic machine deliberately designed by a vanishing civilization. The possibilities range from the supremely simple to the staggeringly complex. At the heart of this entire mystery remains the core philosophical debate we first encountered. Was the genesis of life driven by energy flow, the metabolism first principle, or was it driven by information storage, the genetics first concept? If metabolism came first, then the first living thing was a process, an efficient chemical engine that simply knew how to harness energy to sustain itself, with no blueprint required. If genetics came first, then the first living thing was information, a stable structure like RNA or DNA that mastered self-replication, with energy processing coming later. Many scientists now believe the answer is a compromise, a complex co-evolution where simple metabolic processes and simple genetic codes boosted each other until they finally merged into the first true cell. But the starting point remains fiercely contested. These 30 theories are not just academic arguments about the past. They are the essential guidebooks for our future. Every time the James Webb Space Telescope looks at a distant exoplanet, searching for signs of life, it is looking for a biosignature, a tiny hint of activity. Our ability to recognize those faraway signs depends entirely on how broadly we define life. If we only look for water-based, carbon-copy Earth life, we will miss the methane-based azotosomes on a Titan twin, or the iron-sulfur cycles on a deep-sea world. The deep mystery of abiogenesis is therefore the key to solving the silence of the cosmos. If life began easily and multiple times as a simple chemical process, then the universe is filled with it, waiting for us to find its many forms. If, conversely, life required a complex, specific chain of events, like the perfect synchronization of chiral purity and wet-dry cycling, then we are truly alone. Whether you believe life was a relentless metabolic engine in the planetary crust or a fragile genetic molecule delivered by a passing comet, the search for the universe's first living organism continues. Let us know which theory convinced you. Are you on Team RNA, Team Iron Sulfur, or maybe even Team Panspermia? Leave a comment below with your favorite theory. And if you want more deep dives into the impossible mysteries of space, be sure to subscribe.